Hello students. Now in my previous class I had started with the topic absorption by roots, and I had told you that there are 5 main processes that are involved in this process of absorption by the roots. They were imbibition, diffusion, osmosis, active transport, and the 5th was turgidity and flaccidity. We had discussed 3 in detail, but before I move on to the 4th process, let us quickly revise it in the first few minutes of today's class. First, imbibition. Imbibition is surface adsorption of water molecules because of hydrophilic substances like cellulose and proteins present on the cell wall. So there is surface adsorption or surface attraction of molecules of water. Then diffusion. Diffusion is that process in which the two substances have to be in direct contact with each other and the molecules of a substance move from a region of their higher concentration to a region of their lower concentration till there is uniform distribution of the molecules. So the key point here was that the two regions of different concentrations have to be in direct contact with each other. Third process was osmosis. Osmosis is the movement of water molecules from a dilute solution, or we can say where the concentration of water is high. That is movement of water from its is the key term, its high region to its low region, and this has to be through a semi-permeable membrane. So most important is semi-permeable membrane. The passage of water molecules has to be through a semi-permeable membrane. And we had discussed experiments based on it. I had given you examples of things which show these processes in our day-to-day lives. Now today we will begin with the 4th process, active transport. The word active means involvement of energy. Transport is movement of substances. So in active transport, substances move. Now substances like salts and ions of various ions and salts, like I had mentioned, salts like nitrates, phosphates, and ions like potassium, sodium, chloride, etc. They move from a region of their lower concentration to a region of their higher concentration. That is from outside the cell, from outside the root into the root. This is against the concentration gradient because it is from low to high concentration. So in active transport, the movement is against the concentration. The movement is against the concentration gradient. When I say against, this means from low concentration towards high concentration. And this will not take place without the involvement of energy. So energy in the form of ATP is utilized by the cell, then only movement will take place from low to high concentration. So is that clear? Now the next topic is turgidity and flaccidity. Turgid, if you remember, in the last class I had told you, turgid means when the cell is in a fully extended or distended state. That is, the cell membrane is touching the cell wall. It has absorbed as much water as possible. Now it cannot absorb any more water. The cell is said to be in a turgid state. Now before I explain this, I will explain tonicity to you. Now tonicity is relative concentration of two regions. Relative concentration of two regions, which is going to decide the direction as well as the extent of diffusion. So relative concentration of two regions which will decide the direction and the extent of diffusion. By this I mean there has to be a separating membrane and the two regions, so semi-permeable membrane comes into picture. The two regions, one is going to be of a higher concentration, one is going to be of a lower concentration, or both the regions can be of the same concentration. So depending upon this, we use three terms. Isotonic solutions. Iso means same and tonic word comes from means tension. When there is same tension on both sides of the membrane, that is isotonic, which means the molecules of the solvent, that is water, as well as the solute are same on both sides of the membrane, that is inside the cell as well as outside the cell. So result of this will be no net movement of water molecules. No osmosis will occur. Water molecules will not move inside, water molecules will not move outside the cell, because concentration of water molecules and the solute is same on both sides of the membrane. This is isotonic. Second term is hypotonic. Hypo means less. So when water molecules, I'm just erasing this. Now telling you about, this is suppose this is the inside of the cell, this is outside of the cell. Hypo means less concentrated solution outside the cell, which means more water and the solutes are less. And I have told you it is relative concentration, means we are talking in terms of solutions on both sides of the membrane. So water is more outside, water is less inside, solute is less outside, solute is more inside. The result will be movement of water from where it is high to movement of water where it is low. That is, it is moving inside the cell. This is end osmosis. Inward movement is end osmosis. So if we are talking of a hypotonic solution, means water will move inside the cell, end osmosis will take place. The third term that we use is hypertonic. The word hyper means high or more. Now same thing, this is reversed. Suppose this is the inside of the cell, this is the outside of the cell. This is the inside of the cell, means water inside the cell is more and the solute inside the cell is less. Whereas water is less on the outside of the cell and solutes are more on the outside of the cell. So water always moves from where it is more to where it is less. So the direction here remains the same, but this is inside and this is outside. Don't get confused with this. So movement outside means outward movement is exosmosis. Exosmosis. Exosmosis means movement of water outside the cell. End osmosis, which is taking place in a hypotonic solution, means movement of water inside the cell. Isotonic means concentration is same on both sides, so there is no osmosis. So this is tonicity. So you have to know this to understand the fifth process, turgidity and flaccidity. So we will move on to this. Now, as I have told you earlier, turgid and flaccid are two terms which are opposite to each other. Turgid means a cell is in a fully extended or distended state. Now, And flaccid is opposite of turgid. Flaccid means the cytoplasm inside the cell is shrunken. Now what is a cell? A plant cell has a cell wall like this and a cell membrane inside the cell wall like this. The interior of the cell is filled with cytoplasm. This is the cell wall and inside the cell wall is the cell membrane. Now when it has taken water, the cell membrane is close to the cell wall. The two are sticking like this and there is no chance of any more expansion. So the cell is turgid. In contrast to this, a flaccid cell, this is the cell wall. And because there is less water, so the contents of the cell, this is the cytoplasm and this is the cell membrane. The cell contents are shrunken. We say this is a shrunken or a flaccid cell. So these are conditions of the cell, and the phenomena that leads to a turgid cell is called turgidity, and the phenomena that leads to a flaccid cell is called flaccidity. So have you understood what is turgid and what is flaccid? Now bringing tonicity into picture. Suppose we take three situations. Situation 1, situation 2, situation 3. We put a plant cell in this and the solution outside the plant cell is isotonic. Means water inside the cell, water outside the cell are equal. Solute inside the cell and solute outside the cell is equal. So there will be no osmosis. No osmosis means if water moves through the cell wall and the cell membrane into the cell, same amount water will move outside into the surrounding medium. Isotonic means so the cell will remain in a turgid state. There will not be any change in the cell. In the second situation, if we take a cell and the surrounding solution is hypotonic, which means the water is more outside and inside the water is less. This arrow pointing down means less water, arrow pointing up means more water. So what will happen? The solution is hypotonic, there is more water outside. So naturally water will enter into the cell. And when water will enter into the cell, this will be the result. The cell will be turgid. So if the solution is hypotonic, the cell becomes turgid. In the third situation, we take a cell here. The outside solution is hypertonic. Hypertonic. Hyper means high concentration, which means the solutes are more and water is less. It is comparative. We are comparing the two solutions. And inside the cell, the solute is less and water is more. So what will happen? Water in osmosis moves from a region of its higher concentration to a region of its lower concentration. So water will move out. When water will move out, what will happen? The cytoplasm will become shrunken. So the cell becomes flaccid. So these are three situations when we have taken into account a plant cell. Suppose in place of a plant cell, we take an animal cell. Let us say we take a RBC. RBC is a biconcave cell like this. Isotonic solution, it will remain as it is. In a hypotonic solution, if we take a RBC, it will swell in size like this and become fully turgid. And if too much water will go into it, because an animal cell does not have a cell wall, only cell membrane is present in an animal cell. There is no cell wall which is providing rigidity to a plant cell. In animal cell that is absent, so the plant cell is not, the animal cell is not rigid like the plant cell. What will happen? If too much water enters into the cell, it will get ruptured. It will break. It will get ruptured like this. Rupturing means breaking of the cell membrane. Rupturing of the cell membrane because it does not have a cell wall which is giving it rigidity as in case of plant cells. Now, if a red blood cell is placed in a hypotonic solution, it will shrink. It will shrink because water from the cell has come out. There was more water inside the cell, less water outside the cell. The result is that the water has come out and the cell has become flaccid. So these are three different situations. Now, to explain this further, if we look at a turgid cell, why does water enter into it? Because of development of osmotic pressure. Now, the solutes dissolved inside the cytoplasm. There are solute substances, organic as well as inorganic substances that are dissolved inside the cytoplasm of the cell, which are building up a pressure because of which water moves it. So, movement of water is because of this osmotic pressure. And now, when the cell is becoming turgid, it produces turgor pressure which is applied from inside the cell onto the cell wall. And to counter that turgor pressure, wall pressure is applied by the cell wall on the cell contents. Wall pressure is by cell wall on cell contents. Turgor pressure, this TP, turgor pressure is because of the pressure applied by the cell contents on the cell wall. So, if turgor pressure is equal to wall pressure, nothing will happen to the cell. But if turgor pressure becomes more than the wall pressure, then there will be rupturing of the cell wall. Because the pressure from inside that is exerted on the cell becomes more. So this you must have observed, as I have been telling you that we can observe things around us. Sometimes we see a lemon is split like that. Especially those who have gardens at home. This is commonly seen in tomatoes. When there is excessive movement of water into the cells and the cell wall cannot bear the pressure, turgor pressure from inside the cells, it ruptures. So there is oozing of fluid out. And because of this pressure also, how is this built up? How are the cells getting water? The cells are getting water from the roots. Now I will explain one more thing to you. Let me just erase all this because I need to draw a diagram here. Yesterday I told you the root is connected to the shoot. The xylem of the root is in direct connection with the xylem of the stem. Now the roots absorb water from the surroundings. There are three main reasons for that absorption. One is that the surface area of the root is very high. Surface area is enormous or is too much. Why? Because the roots, there is a primary root, there is a secondary root, there are root hairs, and if you expand them, the surface area is enormous. So all of this is absorbing water. So this is one important feature in a root which is helping in absorption of water by the root. Second, the cells of the root, because root hair are in direct contact with water. What are root hairs? Root hairs are extension of the epidermal cells. Epidermal cells have extension. It's the part of a epidermal cell which are actually coming in contact with water between the soil particles. So the cells of the root hair have a cell sap. Cell sap is whatever is filled inside the vacuole of the plant cell, have a higher concentration cell sap is at a higher concentration than the surrounding water, surrounding solution or surrounding water. This is one more reason that there is less water inside the cell, so more water will move from outside into the cell. Second reason for absorption. So these are some characteristics which the root has, which is facilitating the process of absorption by the roots. Now, once suppose I cut a TS of the root. The TS of the root, suppose this is the TS of the root. And I expand one part of the root for you. The outermost cell is the epidermal cell. And this epidermal cell produces extensions like this, which are called root hairs. Soil particles are there and their water molecules are present in the soil like this. Then we have several layers of cortex with large intercellular spaces in between them. These are layers of cortex. Then we have one layer which is called the endodermal cells or the endodermis. So this is epidermis. These are root hairs. These are cells of the cortex. We call them cortical cells. This is endodermis. And inside we have xylem. These are the xylem cells. In between there are phloem cells. Now this is vas xylem, and xylem and phloem together form the vascular tissue of the plant. Vascular tissue of the root is in direct connection with vascular tissue of the stem. And when I was talking about functions of the root, I told you anchorage or fixation, second was absorption, and third was conduction. So whatever the root has absorbed has to be conducted to the stem. So from the soil it is absorbed by the root hair into the epidermal cell, which moves to the cortical cell. Now more water is present in the epidermal cell, less water is present in the cortical cell. So water from the epidermal cell moves to the cell of the cortex. Then this does not have that much water, so water moves into this cell. As a result, this cell gets deficient in water, so it sucks water from here. So there is a continuous channel that is maintained like this. Enters this endodermis cell and enters the xylem, and from the xylem of the root it moves to the xylem of the stem. So this is building up because of root pressure. And finally it will even go to the leaves. This connection, this is the petiole of the leaf, this is the midrib of the leaf, all that is made up of vascular tissue. So there is a direct connection from root to stem to leaf to branches, and this is helping in transport or conduction of water from root to all parts of the plant. This is a very, very important diagram, and when you solve the questions of the previous year's papers, you will see that this has been asked many times. Generally, there are diagram based questions. They give you a diagram and then they ask you questions related on that diagram. So here they will ask you what is osmosis, label the parts of the diagram, 1, 2, 3, they will write like that, and then you will have to label the part of the diagram. What is endosmosis, what is exosmosis, that is questions related to the topic. So this is one important process that is taking place. So another thing that I want to tell you is, we were discussing turgidity. Now why is this continuous flow or channel of water is maintained? Because when this absorbs water, the root hair of the epidermis, the epidermal cell becomes turgid. But the neighboring cell is not turgid, it is flaccid. So it sucks water. Water moves from a higher concentration to the lower concentration in the adjacent cell. Same way, likewise, moves to the next cell. this becomes flaccid, so it takes more water from there. So turgidity and flaccidity. Now I told you when the cell becomes flaccid, that is the cytoplasm of the cell is shrunken. The cell is flaccid. If the cell remains in this state for a long time, what will happen? All the reactions, you know, cytoplasm is the base or is a part of the living substance of the cell, and all the metabolic reactions are going on in the cytoplasm. So if the cell is in a flaccid state for a very long time, what will happen? The reactions will stop. And if the reactions will stop, what will happen? The functioning of the cell will be affected. And if it is left like that for a very long time, the cell will become dead. That is why you must have seen during daytime, when the cells become flaccid, the leaves droop down. Why? Because during the daytime, what is happening? The rate of transpiration is more than the rate of absorption. So, more water is being lost by the plant and less water is being absorbed by the plant. So, the cells become flaccid and the leaves droop down. The plant parts, the shoot parts, they droop down. We even call them wilting of the plant. But by evening, when there is sunset, when loss of water, transpiration stops, but absorption by the root is continuing. So, water is reaching the cells of the leaf, cells of the shoot, so they regain their turgidity. So, in that short span of time, the cell has not become dead. But if it is there for a long time, the cell will become dead. So, before the cell has become dead, the flaccid state of the cell is turning into the turgid state of the cell. The plant can be saved. Now, when the cell becomes flaccid, we also say the cell has got plasmolysed. Plasmolysed, and the process is plasmolysis. Cell has become flaccid or plasmolysed, and the process is plasmolysis. Lysis means breakdown. Plasmolysis. But before it has got completely plasmolysed and dead and it is put back in water, water is given to the cell, reversal of plasmolysis takes place. Plasmolysis is reversed. Reversal means it goes back into the turgid state, and we use the term deplasmolysis for this. The term is deplasmolysis. The cell has been deplasmolysed. That means before dying, the cell has again got water and is back in its turgid state. Now, this plasmolysis is a very important process, and again, coming back to day-to-day life examples, you all must have had sore throat, and in this season we are getting messages because of coronavirus. There is infection, dryness in the throat, there is infection in the throat, so we are getting advice. Otherwise also we keep getting cold and sore throat. That is because of microorganisms that have infected our throat. So gargling is suggested. What is gargling? Gargling is warm water, hot water with salt in it. When you are adding salt in water, what is it? You are preparing a hypertonic solution. And when you take water and keep it in your throat for a little while, what we call as gargling, what is happening? The microorganism cells are coming in contact with water that has higher salt concentration. As a result, what will happen? Water from inside the bacterial cells will come out. That is, the cells will get plasmolysed. And if they remain plasmolysed for a long time, what will happen? They will die. That is how the number of microorganisms decrease, because so many of them die due to gargling. Another very common example, preparation of pickles. Has anybody seen when pickles are made at home? Now the season is going to come. Mango in the summers, almost every house prepares pickles. Mango pieces are cut, and the first step is lot of salt is put in it and it is kept in the sun. They were dry mango pieces, water was not there. But after a day or two, you see there is fluid all around the mango pieces. From where has that come? Water has been drawn out from the mango cells because of the salt that was outside. The solute concentration inside the cell was less compared to, that is, outside there was hypotonic solution. Water has come out from the cells and now the cells have become dry. So the mango pieces become dry. So if there were bacterial cells or any other spores of bacteria present, they are also dying in the process. So this is a method of preservation of pickles by the process of plasmolysis. Another very common example, and the board even asks questions on it, is that sometimes we see between the crevices of bricks some plants come out because the birds have dispersed the seeds or something, and the roots of those plants are growing in that small soil that has collected over there, small amount of soil that has collected over there. Now if the plant is left like that, especially you must have seen peepal trees growing like that, peepal plant seedlings grow over there because the birds are bringing the seeds. Now if you leave it like that and you don't take out the plant, after some time what will happen? The wall will become weak, it will crack. Sometimes it's very difficult to even pull the plasmolysis of the root cells occur. So the result is de-weeding or removal of that plant that was growing there could be done because it will die. So these are some of the common day-to-day examples of plasmolysis in our surroundings. Now I think I have covered most of the things. I will be giving you questions at the end of this lecture which you will solve, and in my next class I will be dealing with experiments based on all the 5 processes that lead to absorption by the roots, that is diffusion I have already told you, osmosis I have already told you, experiments also I have told you, some we will do regarding plants, and we will go into the applications of these processes in our day-to-day life.